Listo, vamos. Buenos días a todos. Bienvenidos al último día de nuestro congreso. Espero que estén teniendo un agradable tiempo acá en Colombia. Para mí es un placer presentar a la última plenary speaker de nuestro congreso. Voy a hacer la introducción de ella en inglés. Hello. Today we have Dr. Dreta Binford. Dr. Dreta did his PhD with Wayne Madison at the Arizona State University. She is actually a professor of biology at the Lewis R. Clark College in Portland, Oregon, an integrative evolutionary biologist whose work on spider venom evolution has been funded by an NSF Korean grant and by NIH. She serves the Aracnological Society through being the treasurer of the International Society of Arachnology since 2012 and a former president of the American Arachnological Society. She has been profiled in The New Yorker and on Nova, and her love for biology was born growing up as a feral child on a farm in Indiana. She enjoys camping, kayaking, cooking, playing guitar, and singing with friends. Thank you.
Thank you. Thank you all for being here on this last day of the conference. I understand many of you were dancing last night. I look forward to the stories. And I also want to thank the organizers for all their heroic work. It's been an amazing week, and for inviting me here. It's a big honor. I'm going to give this presentation in English because it's in everyone's best interest. If I'm talking too fast, please tell me and I'll slow down. I get excited. I'm going to tell you about a case study focused on Blaxocelacin sicarius and a venom toxin. And it's an origin story of a toxin of significance. But I thought I'd start with my own origin story for two reasons. One, to honor my professor. I'm here in front of you completely because of Ann Ripstra, who, when I was an undergraduate, thinking I would teach high school biology, invited me to do research with her in Tambopata, Peru. And that changed my life in terms of introducing me to the biodiversity, how little we knew, how quickly we were losing it, and my own ability to be a scientist. So I give this in honor of her. And also, I'm just so inspired by the community here and the great mentorship you're doing for each other. And one of my happy jobs as treasurer of the International Society of Arachnology is giving funds to students. And there aren't many students here because it's so early, but oops. But if raise your hand if you've gotten funds, because I want to meet you. I know people's names, but I'd like to congratulate you in person. Okay. So my time in Tambopata ignited a real passion for biodiversity. And spiders, as you all know, there are 52,000 named species. Every time I give a talk, I like to update that. And it's an increase from 272 since the last talk I gave. And that's largely due to work from people in this room. So much great taxonomy is being done. As somebody interested in venom, most of that diversity, them being predators. And of course, venom is one of the primary tools that spiders use. So we've got the species diversity, but in my mind, that's magnified by the chemical diversity that's present in venoms. So this is a model of a single spider's venom. I tried to draw on that to look like a venom gland. A single individual can have hundreds to thousands of chemicals in their venom. And it's a cocktail that includes components that the most famous ones are interacting with the nervous system, so manipulating the nervous system. Some cause cells to rupture, some dissolve the intracellular matrix, some are antimicrobial, some are proteases, but they're these magnificent chemical complexes that are a different scale of biodiversity. So the work I'm going to tell you about is focused on two different scales, the taxonomic scale, which of course we model with phylogenies, but also the molecular scale. So I'll be showing you some gene phylogenies. Sometimes when I give talks, I worry that I show too many phylogenies. I don't worry about that with this crowd, so I'm glad about that. Okay, so the main characters of this talk are spiders in the family Sicariidae, which I think are beautiful in their own right, and the venom toxin, sphingomyelinase D, that has made the sicariids famous among non-scientists. So loxosceles are well known for their bites causing dermatocrotic lesions like this one, and sphingomyelinase D is the cause of those lesions. Also, you all know well that you can people can react to their bites with loxoscelism that occasionally causes systemic syndromes and renal failure. But what we know is that this toxin, sphingomyelinase D, if you make a recombinant version of it, so a pure sample, and inject it into rabbits, you get the full-blown lesions forming. So it's this, it's a sufficient causative agent for lesion formation. What caught my eye as a scholar over 20 years ago is that this enzyme activity was also known in some pathogenic bacteria and fungi, and only from a few loxosceles spiders. So I wanted to know where this toxin came from, and that has been the focus of my work for some time. So before I dive in, I want to just fill in everybody's understanding of how this enzyme works. So sphingomyelinase D is a phospholipase. This is a model of the cell surface. Some phospholipids are sphingomyelin. And this enzyme hydrolyzes this bond here and creates a product that's a cyclic ceramide phosphate. This is something that my collaborator in Arizona discovered in the last decade. So within a single loxosceles or a single sicariid venom, you can have many different paralogs that target different phospholipids. So we call this this family of toxins either PLD toxins or SICTOX, the SICTOX gene family. And I'll come back to this towards the end of my talk. So, but I hope that I want to convince you that the case of the sphingomyelinase D evolution is interesting for a few reasons. Its unique sequence and activity is distinct. It's a poorly known gene family. We called it sicariid toxin because of its presence in Sicariidae. The activity is relatively rare in spider venom, but related to toxins in unrelated organisms. That's cool, right? It causes dermatocrosis in mammals, but that's not why we think it's there. It's actually a potent neurotoxin in insects. So hopefully this video, could you hit the push play and reduce the sound? So, sorry, that's the background noise in my lab. You can see that we've injected purified sphingomyelinase D. You can see this violent twitching of the crickets. So it has a potent neurotoxic effect or myotoxic effect. Okay, so there are 4 chapters that I hope to cover in this talk. I'm going to start by talking about the diversity of this family and some biogeographic history to set the context for comparative work we've done to focus on the origin of this toxin. All right, so most of you, I'm envious because you live in places where there are 6-eyed sand spiders and loxosceles, and they're just such cool animals. So the family Sicariidae has 3 genera: Loxosceles, Sicarius, and Hexothalma. 6-eyed sand spiders, I'm just going to gratuitously show you a video because they're so cool. So they have special hairs on their surface, and they bury themselves in the sand. wait for it. There you go. There's something for me that's just so endearing about that. It's like a puppy or something. When we've had live animals in our lab, if you catch them right after they molt and put them in fine particles, they'll take on the color of the particles. So we've made fluorescent blue sicarius. This one is a fluorescent pink. Just put them in chalk and they'll take on the color. I don't want this to become widespread because I don't want them to be on the market, but they are really just super beautiful animals. Okay. The genus Loxosceles has 149 species, and they're found in the Americas and in Africa, and the rufescens group is in Eurasia. Six-eyed sand spiders. Ivan resurrected the genus Hexothalma in Africa, but they're in just South America, Central America, and Southern Africa. We've been able, my students and I have had great opportunities to travel around the world and sample these spiders from all of their native ranges to try to get an understanding of the phylogenetic structure of this lineage, partly inspired for comparative work. And I just want to thank so many people that are at this conference for helping us in that opportunity. It was life-changing for many of us. And the field work is so fun, and I hope to come back to Argentina and be able to stick my head in holes again and find these animals. This is us in Namibia. Here's a picture of the habitat in Costa Rica. Carlos Viquez was really helpful for us there. You can see a web of Loxosceles that has the characteristic blue sheen with its ribbon silk. So we brought those animals back and did a molecular phylogeny for them that told us a few really cool things. So you have Loxosceles as a result. This is an old phylogeny from 2008. Loxosceles was resolved as monophyletic. So were the six-eyed sand spiders, with Sicarius and Hexothalma as reciprocally monophyletic. But with dating analyses, the splits between the biogeographic structure is very consistent with this lineage being present before Western Gondwana split apart, and dating analyses are supportive of that. And I'm gratified that with deep interrogation of Ivan and others, that pattern has held up. So the genus or the family is quite old, and its current distribution is influenced by Gondwanan vicariants. So since 2008, there have been a lot of exciting advances in sicariid biodiversity and biogeography. It's really fun to see the work that's happening. I'm not going to do that justice, but I encourage any of you to look at the work being done by Alejandro Valdez Mondragon in Mexico, and Ivan and Adalberto, and the many folks who are here. So I appreciate that. One story I want to tell you that we've worked on actively is the question of how and when did Loxosceles arrive in North America? So, given that the origin is from South America, they've of course diversified in North America, and many folks have talked about the Garlandian land bridge, but there are proposed land bridges that predate the modern Isthmus of Panama. One between 75 and 40 million years ago, the Garlandia land bridge between 33 and 35 million years ago. I'll point out that there's a brand new paper that this bridge there's a lot of subtle nuance that's contested, and it's unlikely to be a contiguous bridge, but I recommend Ali and Hedges that came out just a month or so ago. But the distribution that we found largely inspired us applying for funds for CarBio. And so with Ingi Agnerson across many years, we did a lot of expeditions in the Caribbean and adjacent countries. It's really gratifying to see some of that material being used here by many folks. Lots of undergraduates and regional collaborators were able to participate in this work. We have 1,000 great stories. I'll just tell you what we found with Loxosceles. So there's a dated tree that's consistent with Loxosceles dispersing from South America to the Caribbean sometime between 28 and 40 million years ago, and then to North America sometime between 18 and 23 million years ago. And let's see if I can get, so you can see this line that is showing where Garlandia is. And it's pretty clear that Loxosceles has diversified within the Caribbean and it is monophyletic. The reclusive group is resolved as monophyletic with a single dispersal into North America. Those nodes are circled here. And it's what's spectacular. So I want to commend Claudia for the presentation yesterday and Alejandro for their work. Just a month or so ago, published a very beautiful species delimitation analysis from the North American taxa, and estimate that there are at least 55 species and a lot of cryptic diversity that's evolved there, that's diversified in the last 30 to 40 million years. And that just tells us how much cryptic diversity there is in this group to be discovered. Here's a CO1 phylogeny of just the Caribbean Loxosceles that we have with various species limitation methods. We estimate at least 20 species, only 6 of them are named, and the rest are cryptic. So there's much great work to be done still to advance that. Ian Peterson, an undergrad at Lewis and Clark, led a lot of this work. I want to shout out to him. Okay, that's all I'm going to talk about with the sicariidae biogeography, but I hope you're convinced that they're interesting biogeographically because they're old. They originated in Western Gondwana. There's a lot of cryptic diversity. Genetic structure supports the American morphological species groups that were identified by Gertsch and Gertsch-Ennick and Brignoli. And I think that's just fantastic. The taxonomists were able to recognize, despite haplogyne genitalia that don't have a lot of characteristics, identified species groups that are upheld with our molecular analyses. The genetic structure reflects geographic history. And it also seems that rare overwater dispersal events have happened in this lineage with major impact, including the North American radiation. Madivan and colleagues have also demonstrated, done great work on the Galapagos that also shows similar patterns. I would love to just have an invitation. I think this lineage would benefit tremendously from UC analyses, and it would be great to work together on the American fauna. So please talk to me, folks that might be interested in that. Okay. Check my time. So the biogeographic structure of my initial work on that was inspired to set up a framework to ask questions about the evolution of sick tox. So I'm going to transition here to focusing on our work that's demonstrated when this toxin was recruited as a venom toxin. And I'll just tell you, it was a long time ago in a faraway place based on this work. And the way we did this work was to start with the few species that were known, demonstrated to have this toxin in their venom, and just walk phylogenetically backwards until we found relatives that did not have it. And how far did we need to go for that? We were able to bring, I don't want to play this video, actually, no, so don't, oh, wait, stop. Yeah, no, okay, thank you. no video, but we can see this image here. We were able to bring live specimens back from all the regions that we visited, and I was able to obtain samples of venom from them and RNA for transcript, or to analyze which genes were being transcribed. And with that same tree, I've now put a little hot pink star for every species we assayed and detected the sphingomyelinaceae activity in the venoms of them. Every sicariid had some amount of sphingomyelinaceae activity that we could detect with enzyme assays. And the near relatives did not. We had no activity. We've continued scrutinizing this, though, because it's unclear if lack of detection in an enzyme assay is sufficient evidence of absence of this toxin being expressed in other taxa. So it's just a high bar. So we've been characterizing their venoms using venomics, combined transcriptomics and proteomics. This work is, so the key collaborators are from Lewis and Clark, undergrad students, and Pam Zobel-Throp. But to orient you to our taxon sampling, I want to position the sicariids with their close relatives. So their sister taxa include Drymusa and Periogops, some of my favorite species, favorite taxa, and spitting spiders. If we pan out even more, they're in the Synspermiata, which includes a lot of these species here. So the Synspermiata tend to have simple genitalia, haplogyne genitalia. They often have fewer than 8 eyes. It includes falcids, dysderids, digwettids, cytodes. And so we've done venomics, transcriptomics, and proteomics on all of these, on representatives from each of these taxa that has a star. Okay, I'm going to walk through the composition of these venoms, and I'm going to try to summarize some complicated stuff with some in a sensible way. So these are color-coded categories, families of venom toxins that we found across all of these taxa. So for each one, I'm going to show a pie chart in the middle, a phylogeny over here with a star of which taxon it's representing, a picture of the taxon, and then I'll summarize the major patterns. This is partly just gratuitous, so I can show you the venom profiles of these species that are pretty interesting. Okay, so here's a Physocyclus americanus, daddy longleg. You can see the phylogenetic position here. This is the distribution of components in those venoms. They have a large diversity of stase and metalloproteases, neprolysins, and these ICK0 neurotoxins that's cut off at the bottom here. But this big slice of the pie that's hot pink are little peptides that probably are, that are likely homologs of neurotoxins. But astasins and neprolysins are very typical of these. So we published this in an article that basically got some press because of the urban legend that daddy longlegs could kill you if they could only bite you. Turns out they can bite you. It's a little painful, but you're not going to die. That was done with Chuck Christensen, who's just a wealth of knowledge about spider venoms. Importantly, this red component is not there. There's no evidence of sick tox in those venoms. So cytodes. So could you push play on those videos there? The one on the. I have to show videos of cytodes. These are videos that I got from Bob Suter. Cytodes, of course, spit glue on their prey in a very rapid motion. And it's the one on the bottom going to work there. Go ahead and push that one. I love this video. The spider's in the background, a little blurry. It's spitting towards these two metal bars. And you can see the glue that comes out is very contractile and tough. So it pulls those two bars together. A really fun, different model for venom. And here's what their venoms look like. So they're dominated by these little neuropeptides, but they also have a unique family of venom proteins that we've simply called PXCCs. They, when we published this work, had no homologs that were available in databases. And surprisingly, we found one peptide that's homologous to sick tox in their venom. Oops. Okay, so Perigops pseudori also has a lot of peptide neurotoxins. These are the closest relatives we have to sictox or to sicariids. Their venoms look pretty standard for the lineage. Astacins, little peptide neurotoxins, notably no sictox in these venoms. But when we get to the sicariids, you can see this red slice of the pie is sictox. So we have 5 samples across, or 4 samples across the lineage, and work also complemented by work from others for loxosolase lata. But sictox makes up at least 50% of the protein composition of these venoms, and that's true across the entire family. So arguably, something unique happened before the most recent common ancestor of sicariidae within their venoms, and we find these data to be pretty compelling. So if you overlay all of these together, you can see that there's a very distinct and major change in the venom composition within the sicariidae. Okay, so to summarize, we've tested for PLD activity and found across even more species than this, more representatives, and not found it outside of the sicariids. When we look at transcripts, though, we find homologs in every venom gland transcriptomic we've done. We also find homologs expressed in other parts of the body. However, the proteomic analyses, we find evidence of these toxins in the proteome across all sicariidae, but also this unique, weird little peptide that showed up in cytodes. The story was so clean, but then we went and found a homolog being expressed in cytodes. Okay, there's a lot in this figure, but this is a map of the phylogeny with other components mapped onto it. So the components in green are unique to a particular lineage. Components in purple here are unique to loxosolase. Yellow are unique to sicariidae. So we've mapped on all these other peptides onto this phylogeny. And this is notable to us because one of the a priori predictions we had is that when sictox was recruited as a venom toxin, and knowing that it has a neurotoxic effect, we predicted that some of the other components might be lost from the venom, right? But it turns out that's not the case. So these broadly detected gene families that were present before sictox was recruited have persisted. So it seems like with the venom phenotype, evolution can simply add more to it, right? It didn't replace neurotoxins. So there may be synergies going on with sictox and the neurotoxins. We don't know, but phenotypically, venom simply became more complex in the sicariidae. Okay. All right, still doing okay for time. We've gone through 2 of the 4 chapters. How's everybody doing? I worry about the dark space here in the middle on Friday morning. Okay. So I hope you are convinced now that the sictox recruitment is a unique, very cool evolutionary event. So I want to talk a little bit about work I've done with Dr. Matt Cordes from the University of Arizona that's really investigating where this toxin came from. So the deep history of the origin of sphingomyelinase D. Here's Matt with his lovely dog, Aubrey. Okay, so this is a lot of phylogenies for early in the morning on a Friday, but so it's clear that, oh, sorry, the title got cut off, that the sictox gene family is a homolog of GDPDs, or glycerophosphodiesterases. I won't say that word again, but GDPDs. And if you remember from the beginning, I told you that they're also found in pathogenic bacteria and pathogenic fungi, right? Well, it turns out, so we've done extensive data mining in bioinformatics and discovered this phylogenetic pattern. So the green lineage there is actinobacterial. So it's that both the fungal and the bacterial pathogens are found in this green lineage. Other metazoan variants of Sictox are found in what we've called an aquatic lineage, and then this is the sicariid toxin-like lineage. So the fungal and bacterial pathogens are homologs, but they're not closely related to the spider venom toxins. And both of those metazoan lineages have close ancestry or sister taxon relationships with proteobacteria, right? So I'm going to zoom in on this lineage that includes the Sictox. We're just going to get closer and closer to the origin. So this is a complex phylogeny, but I'll point out some of the key points here. So the clade at the top is where the sicariid venoms have been recruited. You can't see those names very well. I can't even see them up here very well, but the arthropod copies are here. With extensive data mining, the closest relatives that we found to the arthropod lineages of Sictox, I was going to have people guess, but I've shown you the answer. Nemertine worms. When we published this work in 2018, the sister taxon was Cnidaria, was corals, right? Now some new sequences from nemertine worms have shown up, and they are the closest relatives to the arthropod expressed Sictox gene family. The only way we can think to explain that was an ancient lateral transfer event, probably in the deep oceans. It turns out that Sictox is present in all the chelicerates. So yeah, consistent with lateral gene transfer in the ancient oceans. That's cut up on the bottom there. I'm going to zoom in on the clade that was circled in the last slide. So this is a phylogeny of all of the Sictox variants that are found in chelicerates. It turns out that, so they're found in chelicerates, they're also found in myriapods. So the most recent common ancestor of arthropods would have had these toxins or this gene family in their genomes. And then it was lost before pancrustacea. With all the genomes in pancrustacea, there are no homologs. So the gene family has an interesting origin and loss story. So this is again a phylogeny of the chelicerates. The presence of these in chelicerates. It's fairly conserved in chelicerates. We found them in all genomes except pycnidogonids. There are 3 clear paralogs in all spiders and 2 or 3 that are present in other arachnids. The non-venom function is unknown, but we are expressing and purifying and testing them and learning some things about their activity. Interestingly, there's limited convergent venom recruitments in what we call the A clade of this gene family. So I've circled here where the venom toxins are, that I'll be talking about in more detail. There's also a homolog that's found in Hemiscorpius and expressed in venoms. And there's a homolog that's expressed in tick saliva, right? So it has been recruited multiple times. I won't be surprised if it's discovered more extensively with more work. Okay, importantly, you can see the 3 blue clades that are the spider variants. Okay, is everybody still with me? I was worried about inserting that, but I just think it's such a cool story. So now I'm going to zoom in on the phylogeny of the venom-expressed variants, and we'll stay there for a while. Okay, so as I've shown you, at least 50% of the venom toxins in Loxosceles arizonica and other sicariids are Sictox variants, and 60% in Leta. it seems that there's been a burst of diversification in the proteins after they've been turned on in the venom. And so we've got evidence of gene duplication leading to new paralogs. As I've just told you, there are, sorry, I'm pointing it in the wrong direction, 2 to 5 copies in the genomes of spiders that aren't expressed in the venoms. After recruitment, there's been an explosion. I'm calling it an adaptive radiation at the molecular level with 23 different variants that are expressed in the venom. So not only is it important functionally, there's diversity in the details of how they're functioning. We found all of those in the venom proteome. Okay. So this is a gene phylogeny for SICTOX. The colored branches represent the venom-expressed clade. So here's the most recent common ancestor of the variants that are expressed in the venom. All of the black branches are not expressed in the venom. Excuse me. We tried to create a nomenclature for this gene family. The alpha clade includes variants that are only expressed in loxosceles. The beta clade they're expressed in loxosceles, sicarius, and hexathalma. We found evidence of some variants that are expressed not only in venom, but in other parts of the body, the same exact variant. So it seems that the toxic activity is contextual. So these variants, some of them are serving other roles in the body. This weird cytodes variant is more closely related to these otherwise body-expressed variants that we find in the sicariidae. All of the well-known clinically relevant toxins are found in the alpha clade that are loxosceles specific. Okay. Commercial break. So that was the summary of the deep history. Hopefully you now know a lot of the fine scale evolutionary history of this toxin. I want to finish this by talking about what we're learning about how this diversification has played out in terms of the functional relevance for envenomation. Again, this is done with Matthew Cordes, but also many Lewis and Clark undergraduates. And to start this, we'll revisit how this enzyme works. Again, this toxin cleaves phospholipid head groups off of cell surfaces. And I think of this as like they basically dock on the cell surface and cut the heads off. And my crude image is like, imagine a lawn where you have dandelions. This toxin comes down and pops the heads off the dandelions, right? But it turns out that cell membranes have different kinds of phospholipids. So there's dandelions and there are crabgrass, right? Some of these enzymes will specifically pop the heads off of the dandelions, others the crabgrass, and others have activity on both kinds. The primary phospholipids are sphingomyelin and ceramide phosphatidylethynolamine. So things that we found that vary in activity, about their activity. So you've got the phospholipid head groups, which are sphingomyelin and lysophosphatidyl-choline, or ceramide phosphatidylethynolamine. And then the phospholipid backbones, the sphingosine backbones and the lysophospholipid backbones. We find activities that target each of these different kinds of enzymes. So the alpha clade tends to have sphingomyelin specific activity. The beta clade is more functionally diverse, so with variable substrates, including sphingomyelinase activity and CPE activity. We've also cloned and expressed these non-venom expressed A-clade genes, and they also have activity on sphingomyelin. So that's not unique to venom expressed variants. We don't know. We've tried for years to figure out what some of these toxins do, and we just don't know. So there's activity that goes beyond what we know anything about. So why are these venoms packed with so many toxins? Well, we know that they are generalist foragers, and their diets include a lot of risky prey. We find them eating each other, they cannibalize each other, they eat ants, they eat scorpions, and they bite and hold or bite and release. So there's another paper, Souza, Silva, and Ferriari that has a beautiful profile of the diets from some cave Loxosceles in Brazil. In 2020, there's the very first record of Sicarius eating a vertebrate. Have any of you seen a Sicarius eating a vertebrate? If there was any group that would, it would be you. So there have been arguments that maybe SICTOX originated to immobilize vertebrates, and that's partly biased by so much of what we know about this toxin is focused on its effect on people, right? But I will argue that the evolution of it has been focused on improving their ability to mobilize insect prey. So yeah, go ahead and hit this video. So I just wanted to show you a little bit of a snippet of how these animals bite prey. This was taken with Hannah Wood with high-speed capability. Amazingly, the spiders, I think all spiders do this, of course, with these tiny chelicerae, they're delivering a bite on the leg. The bite only lasts 1/17 of a second, and then they're releasing and stepping away and waiting for the toxins to take effect, right? When they're attacking different types of prey, sometimes they bite and hold, sometimes they bite and release, but this shows you a little bit of the impact that's needed for these toxins that to act quickly. Okay. So one thing we know is that organisms differ in their predominant phospholipids. So it turns out that crickets have in their cell membranes kind of an equal amount of ceramide phosphatidylethanolamine and sphingomyelin, whereas houseflies are almost entirely CPE dominated, right? We have we're sphingomyelin dominated, and things like C. elegans are sphingomyelin dominated. But so organisms differ in their predominant phospholipids. This is a nice illustration of that. So a table that summarizes the variation of the bulk sphingolipids in arthropods. But CPE is pretty distinctively arthropod. It's not found very much elsewhere. And just to remind you, so we found well, I don't think I've told you this yet, but the alpha clade for Loxosceles is very sphingomyelin specific. That's the one that affects us and causes dermonecrosis. The beta clade, though, has this variable substrate, right? So some of them have evolved to be very specific towards ceramide phosphatidylethanolamine, including a toxin from Sicarius turosus. It's one of the most specific CPE enzymes that is known. Okay, so we've cloned and expressed and purified many of these variants and tested their activity on insects. And so here are some videos. So if you could go ahead and push play on the video on the left. Done is we've taken these purified enzymes, injected them into prey, and basically we see that all of them are neurotoxic on prey. We're trying to quantify differences between the potency of prey that differ and their predominant phospholipids, but it turns out that all of them cause violent, really turgid twitching. Okay, so my students have developed a microinjection assay to test to quantify LD50s and potency on Drosophila. And so here's a picture of how that's working. And this is great because these purified venom toxins are precious and we don't need to use very much material. So here's what we're finding. So the y-axis here is percent awake, and different variants that have different enzyme specificities. So crude venom is a positive control, and buffer is a negative control. Oops. And we're finding that the ST beta, this sicariidae CPE specific enzyme, has higher potency on Drosophila. Not surprisingly. We're also doing cytotoxicity assays on insect cells to see if different variants differ in their effects on insect cells. And they do. It turns out these are difficult assays to wrangle, but we're seeing also higher activity of the CPE specific toxins. So here's just another figure that's showing the differences in cytotoxicity as a function of their specificity on phospholipids. Okay, so we have evidence that's building that the increased CP activity correlates with increased insecticidal potency. We've also done ancestral state reconstructions where we've created hypothetical ancestral proteins and tested their activities, and found that the toxins that represent the most recent common ancestor of the venom-expressed clade and their sister taxon that's non-venom expressed acts on both sphingomyelin and CPE, but prefers sphingomyelin. But the most recent common ancestor of the venom-expressed clade has evolved specificity and a preference for ceramide phosphatidylethynolamine. But then again, within this clade, there's been much evolution of differences in the substrate specificity. Okay, so to answer this question, how has SICTOX diversified and why? There's been a burst of gene duplication with the evolution of functional specificity, and we think that's simply explained, driven by selection to optimize generalist arthropod prey capture, right? No surprise there, but we're having fun documenting that evidence. Okay, so that was a lot. Thank you for your attention. I just want to close by coming back to this origin story of, again, the path that this project has taken has gone to a very deep molecular biochemical level. However, it comes from the very same passion that I have for the evolution of biodiversity across scales. And so this experience of working with the sicariidae has just fanned the flame of that. So some general take-homes that I hope are clear: that sicariid spiders are exciting models for biogeographic analyses, and we're well positioned now to do integrative evolutionary analyses with them. The gene family from which this toxin was recruited has an interesting evolutionary history. And the gene family has gone and undergone a molecular adaptive radiation after recruitment from venom. And the puzzle of biodiversity is magnified by focusing on venom evolution. So with that, there are so many people to thank. The overwhelming majority of them are undergraduates at Lewis and Clark College. I do this work in a very small lab with incredibly talented undergraduates. I'll say that I'm so proud that Huli Cosgrove is here. She started out at Lewis and Clark. And I'll leave you with a picture of their awesome faces. So, thank you. Is everybody still awake? It looks dark. Thank you.
Okay, we have, tenemos tiempo para preguntas, entonces, ya sea en vivo o online.
Hi, Greta. Nice talk. I'm now speculating what could be the functions of those genes and proteins in other animal groups. And I don't know, I can easily imagine them to be digestive enzymes. What do you think about this?
That's a great question, and we're really kind of dying to know. I mentioned early in the presentation that the product of cleavage is something called it's a cyclic ceramide phosphate, and it may be involved in self-signaling. So, yeah, we don't know. But we do know that a lot of the non-venom expressed variants act preferentially on lysophospholipids and not sphingolipids, for example. There's differences in the head group specificities. So there's, yeah, and there's also been some work in Latrodectus transcriptomes that show that they're expressed in early development.
Hi, Greta.
Hello.
Thanks. That was a wonderful story, and I love it all. So you should go to Uruguay, not only to Argentina.
Happily.
Yes. So because I'm more behavioral, I was interested in your experiments. So you say that it could be that the venom evolved for capturing very dangerous prey. So have you tested this type of dangerous prey, like the scorpions? Maybe I was lost with your experiments, but have you tried that? Or
we
sorry,
haven't.
which other dangerous prey can be in the environment?
Well, probably the most dangerous prey are each other. I mean, they're cannibalizing one another. They and eating scorpions. They eat Hymenoptera, pretty much all kinds of arthropods. We have not done, to do careful quantitative comparisons of potency on insects or any target requires a lot of those organisms. I don't have the heart to inject a scorpion, honestly, or I haven't done any vertebrate models because it's hard in the U.S. to do with my lab situation. So we've done some bite removal assays, and we can see symptoms of envenomation in pretty much everything they bite within the first few minutes of the bite. Usually it's pretty quick.
Yeah, but I guess that if you have a more dangerous, I mean, I'm thinking about also the defense of the predator, right? So maybe the venom is very hard for these insects that are not very defendly, but it's not so hard for the very high level defender predators, something like that.
Yeah. So for vertebrates, I may not be answering your question, and just let me know. There's no evidence that the envenomation causes pain or any kind of immediate reaction in vertebrates, which would make it a lousy defensive chemical. Like the onset of dermanocrosis is on the course of hours, right? For a defensive toxin, you'd want it to be acting really quickly. The other neurotoxins in sicariid venoms may be causing pain, but there's no documentation of that I'm aware of. I don't know if anybody in the room is. So, yeah. And so for defense of, when spiders are defending themselves against potential predators, those potential predators become prey. Yeah.
Hi, Greta.
Hi, Sandra.
Awesome talk.
Thank you.
What about the other toxins?
What about them?
I mean, do they evolve in a similar pattern? Like, do they all, I mean, I'm assuming they all come from gene families. Do they have a similar pattern of evolution?
Yes.
Like, what about them?
Yes, we've done some gene tree analyses, some phylogenies of the other toxins. They're also diversifying within sicariids. We've tried to clone, express, and assay them, but it's been hard to recover any kind of neurotoxic activity. There are a lot of astasin metalloproteinases, which are ubiquitous in venoms, right? And a lot of the standard enzymes that are probably breaking down intracellular tissues. But yeah, that's all we know at this point.
Gracias, doctora Greta. Buen día, excelente trabajo. Una pregunta, en el Instituto Nacional de Salud de Colombia estamos desarrollando, pues en búsqueda de desarrollar el antídoto antiloxoscélico para Colombia, pero hemos tenido dificultad en el manejo del veneno, pues por las mínimas cantidades. De pronto, ¿qué recomendación nos puedes dar para obtener grandes volúmenes, sabiendo que es una escala pequeña, de veneno y pues iniciar esas pruebas de potencia con ese material?
He's saying that in the National Institute of Health here in Colombia, they are trying to develop the antivenom for loxosceles, but they are having issues trying to get like enough amount. That if you have any advice.
Yes. Oh, we should talk. Yes, it's challenging. Yes. So sicariidae can give a ton of venom, but loxosceles we usually get less than a microliter per individual, maybe 35 micrograms of total venom protein. So we just pool a lot of individuals. I have no other advice other than I'd love to come and collect venom with you at some point, or we could try to improve your extraction methods, but I bet you're doing everything exactly optimally. So we can also make recombinantly expressed toxin to get more. I apologize for my English, but recombinant expression will get you more protein.
Any more questions? Comment? Online Zoom? No questions in Zoom? Okay. Well, with that, I want to thank you, Greta, for being here today, and thank you, everybody.
Thank you, everybody.
There are some announcements. Wait.
Gracias. Buenos días a todos. Tenemos unos anuncios importantes, por favor. Un momentito. Hoy es la foto oficial del evento, es después del almuerzo, a la 1:30. Por favor, estén atentos a nuestras indicaciones para dirigirlos al sitio donde va a ser la fotografía. Si tenemos evaluadores de resúmenes, por favor, entréguenle aquí a Leonel las evaluaciones, porque necesitamos procesar las evaluaciones para la premiación. Recuerden que los pósters también son virtuales. Ustedes pueden entrar a través del Zoom en el enlace que recibieron. Las personas que no hayan entregado presentaciones, hay varias, por favor diríjanse a la sala rápido a entregarlas, ¿vale? Otra cosa, necesitamos definir la sede del próximo Congreso Latinoamericano de Aracnología, así que las personas que tengan alguna propuesta pueden acercarse para dejar la presentación y hacerla. Y recuerden que hay unos mocks que se están vendiendo para apoyar el fondo de becas Galeano. Entonces, si quieren hacerse a uno, pues nos buscan, por favor, los de la organización. Y las personas que ganaron beca Galeano se pueden quedar ahorita para tomarse una foto con Greta. Greta quiere conocerlos también. Entonces, si hay algunos becarios acá, podrían quedarse de una vez. Gracias. Chao. Disfruten.